This is the beginning prep for lecture 19. Going to start chick development and talk a little bit about mouse development, most likely, depending on the time. And these are similar in that they don't reproduce in water. So the eggs aren't floating around in an aqueous environment. So they're adaptations that allow them to reproduce in the absence of water. And we'll start with the chick, and then, as I said, we'll do a little bit on the mouse, and then we'll finish up the mouse and talk a little bit about human development for the final lecture in this chapter. So the life cycle of the chicken, it's about a 60-day cycle to adulthood, which is pretty quick. Chickens have been well studied in terms of development, although fertilization is internal. The egg develops outside the body. And so after fertilization inside the female, through the oviduct, some of the earliest stages of development occur there. After fertilization, the egg white and the egg shell and so on are put on to the egg so that when it's laid, it's protected from desiccation and so on. But a lot of division has happened already, and so we're going to look at, from a different point of view, this part of the embryo, which is approximately when egg laying happens. So, yeah, so the first 20 hours or so, the egg it's actually rotating around as it's making its way through the oviduct, and that has something to do with anterior-posterior specification, which we will talk about in a later chapter. And then all the divisions are happening, and they're happening in this very small region atop this massive, huge amount of yolk, and then the egg white that's sort of around here as well. A gastrulation occurs, and it doesn't look very much like other types of gastrulation, but it has a number of similarities, and we'll see similarities to the mouse as well, with the node, Henson's node in this case. And so I'll talk about the development of the primitive streak and then Henson's node. It looks like this, actually. It's a case where it looks like there's a little dip in the embryo, and there is a change in cell shape. But what's happening is, rather than forming a flat sphere of cells on the top, some cells are moving inside to form the endoderm and the mesoderm and so on, and the other cells on top are going to continue to form the ectoderm. The somites and the nervous system and all that, they develop just like with the other organisms we've talked about. In this embryo, when the embryos are staged, they're staged by the number of somites at this stage. So there's a 14 somite embryo, a 22 somite, and so on. They form in pairs. And so there are 11 pairs of them, and they start in the anterior, and then they develop more in the posterior. Yeah, and the head process forms first, and so the more posterior structures form later. We aren't going to talk, I don't think, very much about organogenesis, but we probably will be talking towards the end of the semester about limb development. So in these early stages, the limbs are just tiny little buds in the anterior and posterior, maybe not up there, maybe more like here. And then these are elaborated to form, in the case of the chick, 4 limbs that are wings and hind limbs that are legs. And then, we get up to hatching just a few days, 6 days, say, after egg lay. Is that right? No, 21 days after egg lay. And then, it's just growth and maturation and so on. So one of the things that's useful, chickens are not very amenable to genetic analysis, particularly. It hasn't been really a good genetic model organism, but the eggs are available. The eggs are really huge, although the embryos are teeny at the beginning. But you can do this thing called candling, where you can pick the shell off the outside of part of the embryo and shine a light through it. And as long as you keep the membranes intact, you can watch the full development as it's taking place underneath that membrane. And a lot has been learned from that sort of observation. And there's also some other kinds of manipulations you can do, little transplantations of cells and moving them around and so on, which we'll look at later. And there's quite a lot of similarities to mammalian development. And so there's a lot of basic stuff we know from the chick. So when the egg is actually laid, there's, of course, the egg white. There is the big yolk, which is going to feed that embryo as it's developing. There's a little bit of an air sac on one side, and these sort of balancing structures that sort of keep the yolk nicely centered in the egg. It's not, smushing up against the sides and so on. But the little embryo is just a tiny little batch of cells, and it's also referred to as a blastoderm. When the egg is laid, though, so much cell division has taken place that you have something between 32 to 60,000 cells in that embryo. So as I said, these chick and mouse, they don't lay their eggs in water. So we have some issues. We have to have gas exchange. We have to protect them from drying out. And we have to have some kind of environment where the egg is cushioned, the embryo is cushioned and protected the way it would have been if it was floating in water. But wastes have to be stored or got rid of so that they don't cause problems. And so there's two ways of doing that. One is with external reproduction. So the eggs have a porous shell, but there's a series of membranes and so on, and sets of blood vessels that take care of things like gas exchange and the storing of waste until hatching takes place. And then for mouse, it's internal reproduction. So the development is inside the uterus, inside the female, and feeding and waste removal takes place via the placenta. And so the mouse and humans, of course, and other mammals, there's very little yolk. There's a little bit in the egg for those few days before implantation and development of the placenta, but there's not these huge amounts of yolk to contend with. So this is just a schematic. Here's the embryo. I think it's the 40 somite stage. So that's the development. Here's the head and the eye. The embryo is encased in a membrane called the amnion, and this is referred to as the amniotic cavity. So there's fluid in here, and the embryo is inside. When women are about to give birth and you hear the water breaking, this is the amniotic cavity being ruptured so that they can give birth to the baby. So the amniotic cavity is also part of mammalian development. But there's additional structure. So there's a chorion. The chorion is a little thin membrane that just lines the shell. So the shell is porous, and that allows oxygen and carbon dioxide to move across. But you want it to be kind of waterproof. You don't want water to be able to move out because you don't want to dehydrate the embryo. And the allantois is the name of a membrane that extends from the embryo, and there are blood vessels that extend from the embryo to the allantois. This is grossly overrepresented. It's not really this size, but this is where the allantoic membrane comes right up close to the edge where the chorion is and the eggshell, and it mediates the exchange of gas, so oxygen taken in and CO2 released from the metabolic energy of the little embryo. And so the umbilical vein is bringing, is actually bringing oxygenated blood. So the color coding does not correspond necessarily with oxygenation level the way normal diagrams like this would, because we're talking about the embryo. So the the allantoic membrane is picking up oxygen from the outside, and that oxygenates the blood in the umbilical vein, and the umbilical vein then delivers that oxygenated blood to the embryo. And then the the umbilical artery is actually bringing waste products, CO2 and other wastes from the metabolism. And the CO2 is released across the shell membrane, and then the other kinds of metabolic wastes from just metabolism are stored in the allantois, and they are released when the little chick hatches. The other blood vessels are the vitelline artery and the vitelline vein, and the vitelline vein picks up nutrients from the yolk, and the yolk is inside a membrane called the yolk sac, and those nutrients are then brought up and delivered to the embryo, and the vitelline artery returns the blood from the embryo to the yolk sac, and then it picks up oxygen, and it makes connections via capillaries to the veins that then remove the the oxygenated blood. So the oxygenation levels are kind of backwards in the veins and the arteries of the chick at this stage, until they start breathing their own air and so on. Okay, so in the chick embryo, you really have a problem with cell division. So the cells that are on top, over time, they start to divide easily, but the cleavage furrows, they're fine in the place where the actual cytoplasm of the cells, the original cell, are. But when the cells are dividing, the cleavage furrows are not able to penetrate, through the yolk. So you wind up with these cells kind of floating on the surface, and that's why I said, with the zebrafish, and it's similar with the chick embryo. Some of the cells are sort of continuous with the yolk. They don't get actually really cut off. So somewhere on the sides of even the embryo, after it's developed a little bit further, there will be some cells like around these edges that are like this. They're kind of continuous with the yolk. They haven't really separated. So the main cleavage that takes place up here, and this is going to form the actual embryo. So over time, there are a series of divisions, and they lead to a layer of cells up top, and then this little cavity forms. There might be like cell membrane down here, and the divisions just haven't taken place here, and so when a cell division occurs that separates a cell below from above, these ones are all complete, and so they form the blastoderm on top. Okay, and so this little cavity is forming basically just because the cell division planes sort of separate the cells away from the egg, and so this is called the subgerminal space. Sub means below, and germinal. Germ refers to like the embryo. Okay. Over time, as cells divide more and more, if you were to look down on it, you would see this circular, disc-shaped structure, and there'd be the area pellucida, which means clear, and then there'd be the area opaca, opaca, which means opaque or not clear, and this has to do with the density of cells. So the area pellucida, looking from the side, is like this. There's just a few layers of cells, and then the subgerminal space. On the sides, though, you have multiple layers of cells, and so that area just looks darker and thicker. Now, if you take this and rotate it, so this is say anterior, posterior. In this picture, this is anterior and this is posterior. So we're looking on slightly different angles. And so in the posterior of the whole little blastoderm, right in the area pellucida, the cells kind of thicken up here, and this is where gastrulation is going to be initiated. And so at a slightly later stage, we've got a single layer of cells up above and a set of cells. The cells are thicker on the sides, so that's what, you know, the area opaca looks like. But some of the cells in here start to move down and they kind of line the lower surface of the subgerminal space, and they form a tissue called the hypoblast. That is going to be replaced during gastrulation with other cells, but this is a temporary structure. Okay, and one of the first things that you can see just before gastrulation starts is, it's probably not very clear in the diagram, but there's this little crescent of cells in the posterior, and it's thickest kind of at the bottom, and that is referred to as Kohler's sickle because of that crescent moon shape, kind of, and that's where gastrulation is going to begin. And when gastrulation starts, you see a sort of dip. The cells change shape and they form this little groove, and the groove is a primitive streak. So you see it start to form in the posterior, and then it moves. The streak isn't moving, but forward there's signals that tell these cells to start making the streak, and then these cells start making it, and these cells, so that you see this. It looks like this streak is moving, but in fact what's happening is the cells are moving, and it starts in one position, and then the cells up above, more anterior than it, will start doing the same thing. And so there's two things. There's the formation of that groove, and then there's the ingression of cells from this region. They kind of, there's movement. The cells kind of squish together, and they push in this direction, so that these cells in the epiblast actually are going to end up inside, and they're the mesenchyme. They're migrating cells, and they're going to form mesodermal structures. There are also some endodermal cells that move in, and these are going to form endoderm, and they actually, some of them, they start moving in, and they push away the hypoblast, and this forms the endoblast. And then eventually, there's all these different stages, right? But eventually, the endodermal cells are going to move in and push those away as well. So there's you know, multiple there's multiple populations of cells here, and no doubt each one of them is a source of signals that helps to regulate the process. Okay, so this is happening. The primitive streak kind of finishes up at the anterior end, and it gives rise to this structure. This is an area where the cells are kind of thickened a little bit, and it was called Henson's node. And it was called Henson's node because when you're watching gastrulation, it looks like it's moving back. And so I think of this as kind of like, you know, this zipper is forming, and then Henson's node comes along and kind of closes the zipper because the groove closes. And then as this node is quote unquote moving towards the posterior, I'll talk about that in a second, there's this wave of kind of differentiation that occurs behind it. So as it's moving along, the head folds are forming, the neural tube forms, it starts with the neural plate and then the neural folds, and then they move over to form the tube, and then the mesoderm, the somites start to be formed in little blocks as you go along. And so as this zone kind of moves along, it leaves this wake of differentiation. So it's a very important process. Okay, so as soon as we see the Henson's node, we know that gastrulation is done, but then the neurulation and somite and all that is happening, and it occurs in an anterior to posterior direction. And this just shows you, so that at the beginning, the first thing that you see is the head process, and then as the node continues to regress, then you start seeing these later developments. So basically, when the germ layers are forming, I think this should say forming, but you end up with the head fold, but then you get the neural plate, the neural folds, and the neural tubes, and then the somites, and then the digestive system. So there's a series of steps that occur as the node is moving posterior. And it is cells here. These are stem cell-like stem cells, and or stem cell-like cells, and it's called the stem zone. They can only form a subset of structures, and as the Hensen's node is moving towards them and then moves past them, that's when the patterning occurs. It takes still time for the somites to form in the other structures, but the stem cell zone, or the stem zone, kind of is always slightly ahead of the Hensen's node. So there are signals taking place. So the cells themselves are not moving, but the nature of the cells is changing as different gene expression changes occur. Oh, and I thought I had a nice diagram to show that. But so I'll say at this point that I think I've saved that for the actual lecture. But the node is not a physical structure. And the stem zone is not a physical structure. What these are is places where certain genes are being expressed. So you've got certain genes being expressed in the stem zone and certain genes being expressed in the Hensen's node region. And then these cells tell these cells ahead to start expressing the stem zone cells, and the node tells these cells to start forming expressing the genes to act like the node, and so on. So what this is, it's almost like the baton being passed along, just like a relay race. But in this case, we're talking about gene expression. Okay, and so things, the way they're referred to and the way we talk about them, it sounds like it's physical structures, but it really is waves of gene expression that start at one end and move towards the posterior, and then there's other signals that tell, the somites in one region, okay, you're the most anterior ones, and these guys are the more posterior ones. And we have a whole lecture on that later on, so I just want to set the stage. So the neural tube formation of the chick, there's a neural plate that forms. The ectodermal cells on the surface, they get more columnar, they rise up, and they then there's neural folds, so there's a movement of the epidermal cells, the ones that are going to form the skin, and they kind of push upwards, and that creates these like crests, almost like waves, and then eventually these cells are going to meet each other and join, and so then you're going to get epidermis on the top and then the separate neural tube. You can also see there's a notochord here and the developing gut. So if we looked in the chick embryo at a particular stage, we would see the neural plate forming first, followed by the neural crests and the neural tube, and then we would see somites. So these are the somites forming here. Then the notochord, and then the somites, and then the gut. So they develop in a certain order. And if you look midway, you can see that the gut here hasn't formed. It's sort of still open. It hasn't formed the actual gut. But the notochord is here, and the somites are starting to form, and the neural tube is almost closed. And then if we move more posterior, we've got just the neural folds and the neural plate, but we don't have the somites. And we have the notochord here, but we don't have any gut forming at all. And we have certain kinds of mesoderm that hasn't, it's starting to form somites. And then right in the most posterior, you're just starting to get, this is where the node is, and we're just starting to see the folds develop. And so what this means is that as the Hensen's node is moving along, different processes are taking place, and they take place in a certain order. So the first thing is the neural plate, and then the neural tube, and then the notochord, and then the somites, and then the digestive system, the gut. And so that's something to keep in mind. So it's kind of interesting because when you look at the embryo, when it's partially developed, it is like a timeline. You can tell, because Hensen's node moved through the anterior part first, you can tell the order in which these events occur. I think I've just said all these things. So you can, so as I said, the node is an organizing center. It's not a population of certain cells. It turns out that experimentally you could carefully cut out the Hensen's node. And when you do that, the remaining cells, they move in and they seal up the little gap that you've left behind. And there's a little period of time where some sort of communication and reorganization happens. And then a new Hensen's node forms and it just continues on its way. So it means that the node is more like a relationship between cells that are expressing certain genes, and that there's signaling that takes place that sends, induces the cells next door to start expressing those genes while the ones behind stop. And so it's like this little wave. Think of how they used to do in stadiums, the wave, where the people are standing up and throwing their arms up, and then they sit down and you can see, that wave kind of going around. The Hensen's node is kind of like that. And so this is just a summary. From posterior to anterior is the gastrulation process. So this is the primitive streak, and then Hensen's node forms, and it gives rise to the head process. Oh, and the notochord. Sorry, the notochord is before the other things. Yes, because of course, sorry, I forgot. The notochord induces the neural plate formation above it. So then you get the neural plate formation. That's when the ectodermal cells, they get sort of taller and more columnar. And so it's like a thickening. It looks like a thickening, followed by the actual formation of the tube, and then the somites, and then the gut. So that's the order in which those things take place. Okay, and I want to stop there. I think that's an awful lot of material. And so I will introduce the mouse, and I will specifically talk about these things. Maybe some of it will spill over into the next lecture. I'm not sure.